Hi, I'm Professor Seychelle Voss in the Department of Biology at the Massachusetts Institute of Technology. We're standing outside the MIT Lisa T. Su building, also known as the MIT.Nano building. I'm here with my colleague, Professor Joey Davis, also in the biology department at MIT.
Inside MIT.Nano are a series of specialized instruments capable of imaging nano-sized particles like proteins with near atomic resolution. The use of these instruments, which are called cryo-electron microscopes, to image such particles is known as cryo-electron microscopy, or CryoEM for short.
CryoEM can be used to observe the important molecules of life, like DNA, RNA, and proteins. These important molecules are housed within the cell and help to keep the cell alive. In high school biology, we used light microscopes in order to observe organisms like planaria swimming around. But here in this facility, we use electron microscopes in order to visualize molecules that are 10,000 times smaller than that. My name is Dr. Kelsey Miller, and I'm a postdoc in Kathy Drennan's lab here at MIT. And it is my project to study and visualize proteins using CryoEM. Proteins are really the workhorses of the cell. They make things, they break things, they transport things. And my project studies the protein that is responsible for making the building blocks of our DNA. And it is also an important cancer target.
I'm Allison Biester, a chemistry PhD student in the Drennan lab, and I also image protein molecules. The molecules I study allow microorganisms to live on the greenhouse gas carbon dioxide. They use carbon dioxide to make biomass. So I hope that my research can be used to reduce greenhouse gas levels.
And I hope that my research can be used to help make a new cancer drug.
Understanding protein molecules at the atomic level can have many applications both in medicine and in sustainability efforts.
The CryoEM resolution revolution, as it's called, is the result of technological advancements in the design of the electron microscope, in the method for detecting the electron beam, and in the development of new computational tools to analyze the resulting images.
So technological advances are all around us. There are electric cars, there are cell phones, and just like cars and cell phones, the electron microscope has been around for a while. But until recently, we didn't have the resolution we needed to really be able to look inside a protein structure, see the atoms. All we could do is see the overall shape of the protein. I think it looks like the letter V.
When I'm looking for my car in a parking lot, I look for a car with the overall right shape and color. But when my car isn't working, knowing the overall shape is not helpful. Instead, I want to be able to pop the hood and look inside.
So the CryoEM resolution revolution is the equivalent of popping the hood on a car. We can look inside the protein molecule, see what it looks like when it's functioning properly, or see what it looks like when it's not working the way it should. In other words, we can understand the molecular basis of genetic diseases. So I'm Kathy Drennan.
And I'm Krista Emmerich, a graduate student.
And this is the most exciting time to be a structural biologist. We now have the tools we need to make critical advances in medicine and hopefully save the planet.
And we're now back outside of the MIT.Nano building in the middle of MIT's campus. My research group comes to this building to use a very special microscope, the Talos Arctica. This microscope was funded by generous support from the Arnold and Mabel Beckman Foundation. We use this microscope to address a very fundamental question in the cell, gene expression.
Relatedly, my group uses the Arctica to understand how cells build large molecular machines. We're fortunate here in Cambridge to have access to this building because the local environment is subject to tons of vibrational noise. Things like trains and planes and trucks shake everything up, and if this vibration was to propagate to the microscope, it would result in lower resolution images. Fortunately, MIT.Nano is built on a 5 million pound slab of concrete that isolates the entire building from this vibrational noise and thus results in higher resolution images. Okay, let's head on inside.
Hi, my name is Thomas Schwartz. I'm a biology professor at MIT. I'm standing here in the basement of MIT.Nano. MIT.Nano is a brand new building on the MIT campus that was constructed for high-end imaging technology like cryo-electron microscopy. This room is incredibly shielded by the environmental noise, vibration, electromagnetic noise, and it's temperature controlled. We did this in order to house these instruments like this Talos Arctica CryoEM that you see behind me that was funded by the Beckman Foundation. This instrument is used to generate high resolution structures of biological macromolecules, can be proteins, can be protein DNA complexes, can be protein RNA complexes. And with that high resolution, we can explain those molecules much better than otherwise. This is called the resolution revolution. In my lab, we use this for understanding the transport of molecules across the nuclear envelope between cytoplasm and nucleus in a eukaryotic cell.
I'm super grateful for the Arctica. Hi, my name is Nweneka, and I'm an undergraduate at MIT, majoring in computer science and molecular biology. My research involves using data from the Arctica. I get to use my computer science knowledge to develop a structure of a really cool protein that can be used to metabolize components commonly found in crude oil. My research is really important because it would allow us to, in the future, develop more eco-friendly methods for bioremediation.
I'm Andrew, a postdoc.
And I'm Laurel, a grad student.
We're in the Davis lab, and we're very grateful for access to the Arctica microscope.
In some CryoEM facilities, grad students like me are able to prepare their own samples but don't get to participate in collecting the images. At MIT.Nano, however, I'm able to prepare all of my samples and collect the images. This training has been really critical for my development as a scientist. Moreover, a lot of my research focuses on developing new methods for CryoEM, so really understanding how the microscopes work has been really important for that.
CryoEM can provide snapshots of actively functioning molecular machines, like ribosomes, which are responsible for synthesizing all the proteins in cells.
CryoEM can also help us understand how protein complexes assemble. In the Davis lab, we work on developing powerful new computational algorithms for helping us visualize these molecular machines in motion.
On behalf of all the users of the CryoEM facility here in MIT.Nano, I thank the Beckman Foundation for funding this instrument that made it all possible.
So I wanted to add my thanks to the Beckman Foundation. In addition to being a professor, I'm a mom. My daughter, Sammy, age 14, is a freshman in high school, and I worry every day about the world that she will inherit. We need scientific advances now more than ever, and with instruments like the Arctica, we can make those advances. So from scientists and moms everywhere, thank you, Beckman.
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 Thank you, Beckman.